The cell as a living library. Imagine entering a gigantic library containing millions of books. Every book contains instructions for building and running an entire city. That library is the cell nucleus or nucleoid in bacteria. The books are the DNA molecules. The sentences inside those books are the genes. But here's the problem. The master copy of DNA is extremely valuable. The cell cannot risk damaging it every time it needs information. So instead of removing the original book from the library, the cell creates a temporary working copy of the required instructions. The copying process is called transcription. So, transcription is the process by which genetic information stored in DNA is copied into RNA. RNA produced is mainly messenger RNA, which can be abbreviated as mRNA, which carry genetic instructions for the synthesis of proteins. And this is one of the first major steps of gene expression. According to Central Dogma of Molecular Biology by Francis Crick, DNA is converted to RNA and then the RNA is converted to protein. So, transcription is therefore the bridge between genes and protein. So, what we should understand next is why is transcription necessary? Cells do not need proteins, that is, all proteins, I mean, all the time. Like, for example, digestive enzymes are needed only in certain conditions. Stress protein appear during heat shock or under many other stress conditions. Lactose metabolizing enzymes are produced only when lactose is available. Therefore, transcription allows the cell to save energy, respond to the environment, to maintain the balance, and survive changing conditions. You can think of transcription as the cell deciding which instructions are needed right now. So, the main players in transcription are DNA, the information archive. DNA stores hereditary information using four bases, namely adenine, thymine, guanine, and cytosine, and which is normally written as AT and ATGC. Only one strand acts as a template during transcription, and here is the enzyme RNA polymerase, which can also be called as a molecular writer. An RNA polymerase is the key enzyme of transcription, and its job is to read the DNA template and synthesize complementary RNA. Unlike DNA polymerase, RNA polymerase do not require primer and can initiate synthesis independently. And the next term that you should remember while studying transcription is promoter. And this is the starting signal. RNA polymerase does not begin randomly. It recognizes a special DNA sequence called the promoter. The promoter actually acts like a start here, the sign for transcription. In bacteria, important promoter regions include the minus 10 region, which is also called the Pribnow box, and the minus 35 region. And there are various stages of transcription, and the initial or the first step is initiation, which can also be compared to opening the genetic book. RNA polymerase goes and binds to the promoter, and then the DNA strands separate temporarily, forming a transcription bubble. And the enzyme now has access to template strand. This is similar to opening a zipper in the DNA molecule. And the next step is elongation, now, which can be compared to writing the RNA message. RNA polymerase moves along the DNA template. It adds RNA nucleotides one by one. The RNA strands grow in 5' to 2' direction, and the base pairing rules is A pairs with U in RNA and G pairs with C. As transcription proceeds, DNA rewinds behind the enzyme while RNA emerges like a growing ribbon. And the last step is a termination phase, which is finishing the message. Eventually, RNA polymerase reaches a termination signal. The RNA transcript is released, DNA strands reunite, the message is now ready for translation. So there are various kinds of RNA that is produced, and the several forms of RNA that is produced are RNA type function, mRNA carries genetic code, tRNA brings amino acid, rRNA forms ribosomes, and regulatory RNAs control gene expression. Modern biology now recognizes that many RNAs do not code for proteins but regulate cellular activities. This discovery transformed molecular genetics, and there are some modern advances in transcription research, that is, like for example, epigenetics. Scientists discovered that genes can be switched on and off without changing DNA sequence itself. Chemical modifications such as DNA methylation, histone modification affect transcription. This field is called epigenetics. It basically explains the differentiation of the cell, aging, and some diseases, including even cancer. So there are some non-coding RNAs with which previously scientists believed most RNA existed only to make proteins. Now we know many RNAs regulate transcription itself. For example, microRNA, which can be abbreviated as miRNA, and another is siRNA, long non-coding RNA. These molecules can silence gene or regulate development. CRISPR, CRISPR, and gene regulation. The discovery of CRISPR-Cas9 revolutionized genetics. So scientists can now edit genes, and they can even activate transcription or even suppress transcription precisely. And it has various applications, like, for example, in cancer therapy, genetic disease treatment, agricultural improvement, single cell transcriptomics. So, modern sequencing technologies now allow scientists to observe transcription in individual cells. This technique is called single cell RNA sequencing, which helps researchers understand the embryonic development, immune responses, and tumor heterogeneity. So, why should students care about transcription? Transcription is not just a textbook concept, it is connected to genetic engineering, biotechnology, medicine, cancer biology, vaccine development, and personalized medicine. So, for example, mRNA vaccines work because scientists understand transcription and translation mechanisms. So, and when we go towards the transition towards operons, now that we can understand transcription, an important question arises. How does the cell decide when transcription should occur? In bacteria, this control occurs through systems called operons, and there are two classic examples of operon. One is black operon, which is an inducible operon, and the tryptophan operon, which is a repressible operon. These systems demonstrate how cells intelligently regulate transcription according to environmental needs. So, the final thought that we can get in transcription, it is more than a molecular process. It is the language through which life reads its own instructions. Every growth process, every adaptation, every cellular response begins with the careful transcription of genetic information. So, next we will be discussing on detail the regulation of lac operon, what is operon basically, and even the explanation of tryptophan operon. Thank you.